Assalamu alaikum. AppSec's online support program, level 6 to 8, class 8. Textbook Biology 9, PLD Publishers, Lahore, page number 62, chapter number 4, topic cellular structure and functions. Resources required textbook, introduction or summary of the lesson. In today's lecture, we will be able to understand the concept of cellular structures and functions, particularly mitochondria and chloroplast. Army Public School and College Khanewal, Class 8, Subject Biology, Week 14 of FSEX Online Support Program. Topic is 2. Your teacher is Miss Aisha Abid. Let's have a quick recap what we have studied in our previous lectures. We studied about different organelles of the cell, including nucleus and ribosomes. Nucleus is basically the central organelle which is present in both plant and animal cells, and this particular organelle is absent in prokaryotes. Now ribosomes, these are also known as protein factories which are present inside the cell. Mitochondria, these are the double membrane bounded structures found only in eukaryotes. These are the sites of aerobic respiration and are the major energy production centers. The outer membrane of the mitochondria is smooth, but the inner membrane forms many infoldings, and these infoldings are known as cristae. As you can see here in this picture, these infoldings, the yellow one, these are known as the cristae, singular crista. The purpose of this cristae is to increase the cellular, the surface area of inner membrane. This serves to increase the surface area of the inner membrane on which the membrane-bound bound reactions can take place. Mitochondria have their own DNA and ribosomes. The ribosomes of mitochondria are more similar to bacterial ribosomes than to eukaryotic ribosomes. You can see here the structure of mitochondria. It is a double membrane bounded organelle. This is the outer membrane, that one, and this is the inner membrane. This organelle is having its own DNA. The green structure you can see here, the ribosome, and the purple structure, this is known as the DNA. Now you can see here, if we put our focus inside the cell, in the cytoplasm, you can see here these special kind of organelles known as mitochondria. The inner membrane will be having infoldings, and these infoldings are known as cristae. This is the outer membrane and this is the inner membrane of mitochondria. According to our book, the material which is present inside the mitochondria is known as the mitochondrial matrix. This is the cristae, this is the outer membrane, and this is the inner membrane. Now have a look at this video.
Mitochondria. Structure. Mitochondria are intracellular organelles found in almost all human cells. Mitochondria are thought to be derived from the aerobic bacteria that invade the proto-eukaryotic cell more than 1 billion years ago and lived in a symbiotic relationship with it, exchanging energy in the form of adenosine triphosphate for residence. However, this endosymbiotic hypothesis is not universally accepted and has been challenged. Each human cell contains on average hundreds to thousands of mitochondria. The exception is mature red blood cells, which rely exclusively on anaerobic metabolism and contain no mitochondria. Although mitochondria were originally represented as individual isolated organelles, it is now recognized that mitochondria form a dynamic connected network, also called a reticulum or syncytium. Mitochondria have 4 main compartments. The outer membrane, which is permeable to certain ions and small molecules. The intermembrane space, which has a composition similar to that of cytosol. The inner membrane, in which are found the respiratory chain proteins. The inner membrane is folded into multiple cristae, allowing for a large surface area. The matrix, or the inner part of the mitochondrion, where most of the metabolic reactions take place. The mitochondrial DNA is found within DNA-protein complexes called nucleoids in the mitochondrial matrix. Function. Mitochondria are intracellular organelles that are essential for aerobic metabolism and energy production through oxidative phosphorylation, which is accomplished by the respiratory chain. However, mitochondria are not only the ATP-producing powerhouses of the cell. Mitochondria are involved in several other metabolic pathways, including beta oxidation, the Krebs cycle, and the synthesis of iron-sulfur clusters. In addition, mitochondria maintain, replicate, and transcribe their own DNA and translate mRNA into protein. The import and assembly of proteins is another important mitochondrial function, since most proteins that the mitochondria require are encoded by nuclear DNA and translated in the cytosol. The ongoing remodeling of the mitochondrial network is also a function of the mitochondria. Additional roles of mitochondria in apoptosis, production of reactive oxygen species, calcium homeostasis, maintenance of lipid membrane, and immunity have been described.
Our next topic will be plastids. They are also membrane-bound organelles that occur in the cells of plants and photosynthetic protists, particularly algae. They are three types: chloroplast, leucoplast, and chromoplast. Like mitochondria, chloroplast is also bound by a double membrane, one outer membrane and inner membrane. Outer membrane is smooth, while inner membrane give rise to sac-like structure. These are known as thylakoids. The stack of thylakoid is known as granum. You can see here, this is the green one is the outer membrane, the purple one, here it comes the space between these two membranes. Inner membrane forms the infoldings known as thylakoids. The stack of thylakoid is known as granum, singular grana, and the space between these thylakoid is known as thylakoid space. You can see here the outer membrane, the inner membrane. This is the inner membrane and this is the granum. This one whole stack of thylakoid is known as granum, and this is the stroma in which these thylakoids are present. Grana, they float in the inner fluid of chloroplast known as stroma. Chloroplasts are the site of photosynthesis in eukaryotes. They contain chlorophyll, the green pigment necessary for the photosynthesis, and associated pigments. These pigments are present in thylakoids of grana. The second type of plastid in this plant cell are chromoplast. They contain pigments associated with the bright colors and are present in the cells of flower petals and fruits, etc. Their function is to give colors to these parts and thus help in pollination and dispersal of fruit. Leucoplasts, these are the third kind of plastids. They are colorless and store starch and lipids. They are present in the cells of those parts where the food is stored. You can see here the plastids. These are the storage sacs in plant cells only, and the structure is there are three main different types of plastid. One, chromoplast, which will be storing different colors, due to which flowers appear as a different color that we see: red, pink, yellow, orange. And chloroplast, it is having a green pigment. With the help of these chloroplast, plant can do photosynthesis. In chloroplast, there is chlorophyll green substance present. The leucoplast, these are the storage organelles that help in the storage of lipids and starch as well. Let's have a look at this video to understand the concept.
Let's take one look at a common generic plant cell. It's distinctive because of the cell wall. Now, I brought in a plant cell because there is another organelle with the same amazing history as the mitochondria, but this organelle is found only in plant cells. It is that major organelle where the super amazing food making process of photosynthesis actually goes ahead and happens. The plastid. The story is the same. The plastid existed as a type of bacteria really long time ago. One day a plant cell came along, absorbed one of these, realized that it would be an awesome bacteria to actually keep inside it. It converts light into glucose compounds. No one knows how this happened, but scientists know for sure that it happened because both mitochondria and plastids have double-layered membranes, one from its own and one from the cell which actually went ahead and engulfed it. So plastids are of three types: leucoplasts, chromoplasts, and chloroplasts. And chloroplasts are the most important of them. And what they do is they convert light into sugar and oxygen. The plants don't need the oxygen, so they get rid of it and give it to us to actually go ahead and breathe it and then be alive. All the green parts of the plant that you see, the non-woody stem, the leaves, the unripened fruits, all of them contain chloroplasts. So we'll get into more detail about a chloroplast. It looks something like this. Two membranes protect the inner parts of the chloroplast. They appropriately named the outer and the inner membranes. So the inner membrane surrounds the stroma and the grana. The grana are these stacks. Stroma is a kind of cytoplasm or matrix of the chloroplast. It contains enzymes required for the synthesis of carbohydrates and proteins. So one stack is called a granum, which contains many thylakoids. So chlorophyll molecules sit on the surface of each thylakoid, capture light energy from the sun, like sunbathing. As energy-rich molecules are created, they move to the stroma where carbon can be fixed and sugars can actually be synthesized. So this is how the process actually happens. The stacks of thylakoid sacs connected. Can you see these connections? These are called the stromal lamellae. This is also called the skeleton of the chloroplast, keeping all of these grana safe distance away from each other, increasing the efficiency of the organelle. So imagine that they were all bunched up together, lying on top of each other. It wouldn't be efficient way to capture the sun's energy, right? So the lamellae do this neat, I'll say arrangement work for the chloroplast. In case you did not know how photosynthesis happens, I want to run you down by that once. The purpose of the chloroplast is to make sugar, to feed the cell's machinery. Photosynthesis is a process of a plant taking energy from the sun and creating this energy in the form of sugar. When the sun energy hits the chloroplast, typically the chlorophyll molecules, what I'm talking about, light energy gets converted into chemical energy found in components like ATP and NADPH. Now, these energy-rich compounds move into the stroma where the enzymes fix the carbon atoms from carbon dioxide. The molecular reactions eventually create sugar and oxygen. Plants and animals then use the sugars for food and energy, and animals also breathe the oxygen that is released. Okay, so I hope it's really clear in case you did not know how photosynthesis happens. So chloroplasts have many functions. One obvious one, which I've been bombarding you with again and again, is photosynthesis. You know it, so I'm going to repeat what it is all over again. The second thing is provision of oxygen supply. Chloroplasts provide oxygen, the byproduct of photosynthesis, to all aerobic organisms for respiration. This also we actually just went through. Another interesting thing is that chloroplasts temporarily store starch grains in something called pyrenoid. At night, the starch is transferred to regions of growth and storage. What else a chloroplast does is it utilizes carbon dioxide and it fixes carbon dioxide, keeping the concentration in the air, to its normal level. Chloroplasts can also, change into chromoplasts and give you a lot of color in flowers and fruits and, actually go ahead and attract animals. And also fat droplets are stored as plastoglobuli in the matrix of chloroplasts. But still, these are all secondary functions. The primary function of the chloroplast remains as the energy generation through a process called photosynthesis. I hope it's really clear.
So that is all about chloroplast and mitochondria, and we studied about different types of plastids as well. Now your assignment for today is that you have to write down the difference between the different types of plastids, which we have learned that there are three main kinds of plastids. Then you have to describe the structure and function of mitochondria. You have to draw the labeled diagram of mitochondria as well, showing outer membrane, inner membrane, matrix, and cristae as well. You can also show DNA and ribosomes in this structure as well. So in order to differentiate between different types of plastids, you have to you have to do it in the tabular form, mention each types and all of these main characteristics. And this is your assignment for this week. This is all for your today's lecture. If you have any question regarding this lecture, regarding any of the lecture that we have studied, feel free to ask. Thank you so much for your attention. Allah Hafiz.